So plant cells. I'm gonna go through the organelles that we haven't covered in animal cells mostly, and yeah, we'll take it from. So the cell wall, it has an outer layer that maintains cell shape and protects cells from mechanical damage, made of cellulose, other polysaccharides, and protein. You have the plasmodesmata, which are channels that goes through walls and connects the cytoplasm of each adjacent cells. The chloroplast, a photosynthetic organelle that covers energy of sunlight, like that converting energy of sunlight to chemical energy and stores it into sugar molecules, amongst other molecules. The central vacuole, that's a prominent organelle that in most plant cell it functions like a storage, breakdown of waste product, hydrolysis of macromolecules. Enlargement of the vacuole is what makes it like well the cell itself become a bit bigger, and yeah, it is the major mechanism of plant growth. And what are in plant cells but not animal cells are the chloroplast, central vacuole, the cell wall, and plasmodesmata. um so any questions? Slide 11, JPL.
I already changed the slide.
Okay, then it's me. There you go, I see it. Okay. So the polysaccharide in the plant cell wall have evolved very differently from the animal eukaryote. So probably due to a lower need of being mobile and favoring a structural strength instead. They have about 80 cellulose molecules meshed or associated together to form a microfibril, which are stacked and wound in layers to secure a robust cell walls. Mm, The parallel cellulose molecule are held by hydrogen bonds between the hydroxyl groups and polymers of unbranched beta glucose monomers. Um, but yeah, we could also we will precise afterwards how they differentiate, because you have cellulose, you have hemicellulose, and lignin, which are I think it's 80% cellulose, uh, sort of 19% like, or 18% hemicellulose and 2% lignin, something like that. It varies from cell type to cell type, but that's mainly it. And actually, yeah. Any questions? Okay. Um, Slide 12. So the chloroplast. It is enclosed by two membranes separated by a narrow intermembrane, which is where photosynthesis photosynthesis takes place. The stroma surrounds a third compartment, namely the telocherate space, delineated by the telocherate membrane. The telechoids are interconnected as telechoid sacs that are stacked to form structures called grana. Singular grana. Bear with me here, because like we're gonna have like a brush up on how to spell those words and so on with Gabriel, with Latin and Greek um, courses. So yeah, we're gonna get better at pronouncing those terms. But for now, yeah, I'm doing what I can. Which those are further connected by thin tubules between individual telochroids. Photosynthetic enzymes are embedded in telochoid membranes, and free ribosomes are present in stroma along with copies of the chloroplast genome. A bit like the yeah, a bit like the mitochondria. They also have like ribosomes inside that basically like secures the entire like genes and shape of it. So any questions so far? Slide 13. The water transport in plants occurs from evaporation, that where the leaves pull the water upward. The root, the root through water conducting cell utilize diffusion and osmosis. So it pulls water uh, towards them from the ground and pushes it upwards to the various branches and leaves. The properties of cohesion and adhesion allows some of the tallest trees to transport water for more than 100 meters upward. Questions? Cool. Um, Slide 14. So we have prokaryotes. They have like a cell wall. That's the structure differs in gram-positive and gram-negative bacteria, which we'll cover shortly afterwards. The fimbriae, those are like the small hair-like fibrils, helping the cell to adhere to other cells or to a substrate. Some will also be more specific and help them reproduce, but the tunneling electron microscope. Okay, no, okay, yeah, basically, the capsule, sticky layer of polysaccharide. So this is like basically like yeah, a bit like plants. They have uh, polysaccharides or protein that help adherence and evasion. The plasma membrane, no nucleus or other membrane bounded organelle. So it's like it has no shell. Like the DNA is basically aggregated in the middle, but is not like protected by any shell, or it's just a bunch of spaghetti that lays in the middle. uh, so we have the flagella. Those are organs used by most motile bacteria for propulsion. Many species can move towards or away from certain stimuli. And the nucleotide, that's more specific to what's inside the middle. Like they have circular chromosomes, which are plasmids, but also the DNA itself, the noodle itself in the middle is actually also circular. Um, so yeah, it's all circular DNA inside of it. Any questions so far?
So this circular DNA, this is where we start getting into the concept of plasmids, right?
Mm-hmm.
Yeah.
But let's cover slide 15, JPL. Let's cover the gram-positive and negative bacteria. So this is a method called staining, called gram stain, named after a Danish bacteriologist, Hans Christian Gram, is used to distinguish and classify bacteria into two large groups, namely those gram-positive and gram-negative. The staining differentiate bacteria by the chemical and physical properties of their cell wall. The technique is used to make preliminary morphologic identification, and it's worth to note that some organisms, amongst, like they can be E. coli, like E. coli can be gram variable. Like some organism can be gram variable. That means that it'll stain variably on the same cell sample. It means that for some reason, some E. coli, for example, will have different cell wall structure of the same genome, same cell sample, but sometimes they just differentiate in different ways. So you have the gram-positive cell wall. It contains a thick layer of peptidoglycan, and it retains the primary stain crystal violet. Um, cytoplasmic lipid membrane and teichoic acid and lipoids, forming the lipoteichoic acid, which serve as chelating agents. Basically, like a chelate is a inorganic ionic binding of from an ion, like a metal ion, and an organic molecule. So so that is helpful for dissolving metal cations, and also for a certain type of adherence. Back to we'll cover that afterwards. But you have the amino acid called histidine that has this aromatic imidazole, and that is helpful for either adhering to inorganic molecules or also dissolving them. You have gram-negative cell wall. Those contain a thinner layer of peptidoglycan and allows the crystal violet stain to wash out on addition of ethanol. They're after stained pink or red by saffron or fuchsin counterstains to be able to see them. Any questions so far?
Yes. So you said that some bacteria can be variable.
Yeah,
Um.
your
So
your gut bacteria, for example, E.
so
coli can be gram. Yeah.
so for example, does that mean that it switches between one and the other, or it just or it has to be a different bacteria for it to be a different type?
Well, JPL, correct me if I'm wrong, but I think it's from what I read is when they differentiate, some will basically adopt a gram-positive or gram-negative structure. It would like basically stack more peptidoglycan than others, and that's in the differentiation. That's when they have like daughter and daughter cells.
Okay. Thanks.
No
Ha-
problem.
has anyone else lost a stream from JPL?
Yeah.
Oh,
Damn it.
it's back.
Okay. Yeah, I see it too. Okay. Slide 7.
I'm just gonna just double.
Yeah.
So I think we lost JPL again. But also,
JPL, are you there?
yeah, he seems like he's blipping out.
Okay.
Gram positive and gram negative are usually species specific, as far as I know. Like you
Yeah.
don't have an E. coli that differentiates into suddenly becoming a different type of gram stain. But
Okay.
yeah, I haven't studied this in a while, but that's
Well,
what I remember from my microbiology class.
Me and JPL, we shared an article where this could happen. Actually, JPL, you there?
Can you guys hear me? I
Yeah,
had
now we can
like
hear
the
you.
Yes,
most
we
really
can
clear
now.
hiccups.
Okay.
So basically,
yeah.
the thing that actually talks about the gram variability is actually when the strain of bacteria. So, for example, an extraordinarily bacterial organism is actually cannot be, because of its very specific physiology, can't show clear results to the gram staining.
Okay, but this doesn't answer the question, which
Yeah,
is,
yeah.
can I am under the understanding that like E. coli, for instance, they're gram negative. They're always gram negative. We're not going to get like hot swapping E. coli that are suddenly becoming gram positive as they
That's what we talked about,
differentiate.
JPL. But okay, maybe I don't know. I should go back to the article we kind of read. It was basically it could happen that they would change, like, but yeah, okay.
It sounds like a
Let-
highly specific circumstance. Gram
let's take
staining
it
is
is. I think it is, yeah.
species specific.
Okay, let's stick to Gabriel's word on it, and that they're always gram negative. Right?
Yeah.
Cool.
For all intents and purposes.
Right. Some bacteria can be more variable than E. coli, I guess, or like I don't like let's put a pin on that. I'll come back to it. but yeah, for now, E. coli gram negative, and yeah. Any further questions? Okay. Um, slide 16, JPL. So archaea, that's like Ali. You were asking what is an archaea at all, and I did my best to look it up, and those are nice little species of archaea, and the way they differentiate is like, but initially they were classified as bacteria, but despite the visual similarity, they possess gene and serve several metabolic pathways that are more closely related to eukaryotes.
Does anyone else not see the slide?
I see them. I see JPL slide. You may want to reload, basically like a
There we go. Yeah,
yeah.
it's
I
back.
see it now.
Okay, cool, perfect. Yeah. So yeah, so they have metabolic pathways, means that their proteins and RNA DNA will basically function a bit more like our type of cell, like the plant cell and animal cell, than the bacteria type of cell. That's what differentiates them from bacterias, and it also kind of is a big hint on bacteria and archaea mm, uh, symbiotically fusion together to create us. And so the archaea use more energy source than eukaryote, though, such as like metal ion and even hydrogen gas. They can tolerate high salinity concentration, use sunlight, and absorb carbon, although they actually don't do both. Like cyanobacteria, like no known archaea species both use sunlight and absorb carbon like plants. They do either or, like they reproduce asexually by binary fission, fragmentation, budding, and unlike bacteria, no known species of archaea form endospores. I highly recommend you to look this word up, as I would not be able to be more specific on what exactly is fission, fragmentation, or budding reproduction. I can look at that up if you really want me to, and we can talk about it later. But yeah, basically they reproduce in different ways. They use more energy sources than eukaryotes, and the enzyme involved in transcription, translation, and the lipids in their cell membranes are still closely related to bacteria, though. Yeah. So yeah, to finish that's why they're also called extremophile, because they tolerate like they can use hydrogen gas and metal ion to convert energy. They can live yeah, a lot of places and so on. Any questions? Okay. Slide 17. JPL. I don't see anything change. Do you see anything change?
No.
Mm, okay. Should we have another one displaying those slides? You want to do it, Ollie, or Gabriel? Maybe, I don't know. Yeah, in
Sure.
11 days. Yeah.
Give me a moment.
I will. Cool, cool.
Go over. Right.
He told me he reinstalled the whole system and everything, so we were believing in it, but it happened again.
Oh, okay, it does that. There we go. Can you guys see it?
It's loading.
JPL, can you turn yours off, or are we just going to do multiple screens?
Yeah, no, you should click on Ollie Fellow's stream now and watch it full screen if well, not full screen, but Discord full screen.
So mine's working.
But yeah, I see it. Can you switch to slide 17? Perfect. Okay. You guys all on slide 17? You could also basically download the slide document, but yeah, it's working for me. It should be working for everyone. So last check up. Is there anyone not following on the slides right now? Perfect. Cell fractionation through centrifugation. This is a method used to isolate cell components based on size and density. First, the cells are homogenized, which means they're either suspended or mixed in a way that they're diffused equally.
Hello?
I can't hear him.
Hello? 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 Can someone hear me?
I can hear Dr. JPL now.
Okay.
But
JPL, can you turn off your stream? We're trying to get one stable thing working. And Anarchy is
dead.
gone. JPL, do you want to pick up where he left off?
He prepared the slides and everything. I'm normally only here for the Molecular Biology Club to present the slides. I have no idea what he plans. I talked to him that he should reconnect.
All right, well, we'll see how it goes.
Is anybody opposed to using Zoom for this?
Only that we already
Yes.
have everything in
Only-
Discord.
Yeah, only in as much as that, one, everything is in Discord. Two, if the problem is internet connection,
Yeah, that's
switching
not
to a
right.
different thing that's not really going to change anything.
Okay,
Right.
well, I was just gonna throw it out there. I've got a corporate Zoom, and I can load up to I think 500 people in a room. So it's an
Well,
option.
that doesn't actually sound terrible, but oh, there you find it. Oh, he's back.
Can we hear you, Soizek?
You
There we
gotta
go.
love Discord, right? Okay, where did I left to?
Basically at
when
the
did
start
I-
of this slide.
Okay,
Still
perfect.
there. Has been a while.
Homogenizing the
Yeah.
cells. Or...
All right, I'll go through it again. So basically, cell centrifugation and fractionation. So let's go through it. You start with mixing them, and then you centrifugate them at different at time specific force and duration. So you're gonna have the first one's gonna be like the DNA material, like the nucleus. The second one's gonna be the mitochondria, like the mitochondria and chloroplast, the energy production. Then you have the microsome, which are like pieces of plasma membranes and internal membranes. And last, you're gonna have the ribosome, those that produces. So in early experiments, researchers used this type of technique to further in the next slide, use it in microscopy to determine what were cell made of, and that is what established the baseline for today's method of differentiation. So yeah, any questions so far?
So I do have one comment. I know that this is a tremendous amount of information that Aniki is putting forth here. And I know that everybody is either kind of listening with one ear, listening with both ears. Hopefully people are taking notes. But if you have, if there's something you hear and you're like, I have no idea what you're talking about here, if you're too uncomfortable to speak up now, which is fine, you can at least write something down, and then you can message me, or you can message Aniki, or you can message JPL, and all of us are going to have a little bit more information that we can get to, and also you can just Google stuff. So don't feel overwhelmed. Just kind of rack it up in the background and let it kind of sit there and marinate. And then if you need more help, please reach out. Okay.
Right. To add up on Gabriel's statement is that, yeah, Googling is an art. Like looking up terms is an art. And some will be more efficient at finding information than others, but still, like, it would be nice to have you making a little attempt. And if you really cannot find anything after 2 or 3, like if you think, okay, this is too much, please reach out to either me or Gabriel or JPL, and we'll be happy to answer any question. On top of that, we like I would advise now is also to maybe look for the specific channels in Sci-House Discord, because usually there is a lot of information pinned and put in there, especially in the No Noodle channel that we strive to keep at least yeah theoretical. But anyways, so any further questions? Okay, I'll have them. Well, yeah. I heard that.
I just want to ask what I missed at the very beginning of the course.
Oh, we went through plant cells, and well, you can see that we started off at plant cells, and the rest is pinned.
You can work through the slides, and it's pinned on a drive as well.
So can I have the slides document or folder or?
Yeah,
right.
go to
It's in the library.
Go to Plasmid Creation Club, and it's pinned.
It's microbiology crash course.
Hey, thanks.
We should probably make a channel for it, but yeah, it's in the Plasmid Club for now, and it's in the pins. If you look at the pins, it's like we have both the video of the first part, which was from evolution to plant cells, and then we're taking it up from plant cells to light and electron microscopy, which is the last slide. We're gonna attack the last slide right now, and I'll be happy to answer any questions, Sasha.
Thank you.
No problem. Slide 18, or unless there is more. Anything? Light and electron microscopy. Once a specific organism, organelle, or multicellular mix of interest is assumed to be isolated enough through different means of separation, various techniques are then used to visualize the development of expected result in experiment. These are only a few examples of microscopy, but more exist with varying difficulty to obtain. I don't know what's happening. I don't English well today. So, but anyways, so yeah, I will let you guys go through it, or actually, I'm gonna go through it. Bright field, unstained specimen. Passes light directly through specimen unless cell is naturally pigmented or artificially stained. The image has little contrast. Parts AD shows a human cheek epithelial. So as you can see, it's very important to isolate specific cells, else light is going to go through many of them, and you're not going to have like the specific cell that you want to study. So it's important to have like as few of them as possible in order to have basically a one-dimensional view of it, even though it's not one dimension. But like, you don't want one right behind or right in front of each other, else it's gonna clog up the result. So B, bright field stain specimen. Staining with various dyes enhance contrast. Most staining procedures require that cell are fixed, observed. C, phase contrast. Enhance contrast in unstained cell by amplifying variation in density with specimen, especially useful for examining living unpigmented cells. Differential interference contrast, Normarsky, which is the name of it, like phase contrast microscopy, uses local modification to exaggerate differences in density, making the image appear almost 3D. Fluorescence microscopy shows the location of specific molecules in the cell by tagging the molecules with fluorescent dyes or antibodies. These fluorescent substances absorb ultraviolet radiation and emit visible light, as shown. Confocal, a fluorescent optical sectioning, the technique that uses a pinhole aperture to eliminate out-of-focus light from a thick sample, creating a simple plane of fluorescence in the image by capturing sharp image at as many different planes. Okay, this book is amazing, but yeah, I did a lot of effort to have it more verbose per se. So I probably should have made a little talking paper out of that, but it's also just to show you guys how it is to read, basically, right? It's also why I took many of the description out of the slides and in the talking paper. So I want to motivate you guys to read more and not being afraid of reading a lot, because your brain is just a muscle. It's going to get used to it. So in the nanotechnology world, we have scanning electron microscopy and transmission electron microscopy, which are more about how you. It's more quantum physics at this point, but let's focus on what it does. You have micrograph that takes a scanning electron microscope 3D image of the surface, and this image shows the surface of a cell from a rabbit trachea. So like, yeah, it's covered with motile organelles called cilia. The beating of the cilia helps move inhaled debris upward towards the. So yeah, that's if you're a smoker, that's the one organelle that you're basically damaging. So don't smoke, kids. It's bad. So the TEM, transmission electron microscopy, is it profiles a thin section of a specimen. Here we see a section through a tracheal cell, revealing its ultrastructure. In preparing the TEM, some cilia were cut along their length, creating a longitudinal section, while other cilia were cut straight across, creating cross section. And yeah, that's again, it's a lot to take in with the nomenclature and words, but don't be afraid. Your brain will sort out the information and keep what's relevant for now. I mean, believe in your brain, guys. Y'all can do it.
Just even this is only a smattering of what's out there for imaging techniques.
Exactly.
There's a bunch of things missing off of here. There's 2-photon. There's XRM, which is X-ray microscopy. There's
one of the
tomography.
latest. Yeah. One of the
Out of
latest.
curiosity,
Yeah. Sorry.
out of pure curiosity, is the 2-photon one the one that Justin addressed in his stream? The
Yeah,
latest
the 2-photons. Yeah, it's very expensive, but the images it produces are amazing. Yeah.
So with 2 infrared photons making a ultraviolet one.
Yeah, basically, yeah. So it's more selective, and you can pinpoint what you exactly want to look at. It's amazing.
Thank you.
But yeah, those was like raw examples, and it's also an example on how the literature is framed, and how it's easy to go through it. Just let your brain absorb some of the words it wants to absorb, and 30 seconds later, when it goes into your lung memory receptor of the brain, it's all like sort of sorted, and then like tomorrow when you sleep on it, it's gonna be even more sorted. So believe in it, guys. I believe in you.
One thing I noticed from like looking, and I'm not a microscopist, but I work with them. One thing I notice is microscopy is one of those fields where you have to have knowledge in a lot of different areas. There's a lot of physics, optics, chemistry, biology. I mean, it's a really good way to get your toe in a lot of waters. So
yeah, exactly. So is the brain cheese project. Like you gotta go with like programming and machine learning almost, and it's you don't be afraid of changing a little bit of fields to have a more um and ambivalent kind of understanding of things. So yeah, any questions before we wrap it up? Can you guys hear me still? Hello?
yes,
Okay, cool.
Yeah, we can hear you.
Good.
So is this the end of the slides?
It's the end of the slides. I thought we would be talking a little bit more, especially with the
So any of you who have just like I think one of the questions that should be asked, and so I guess I'm going to ask it, is we're trying to get to plasmid creation, right? Like the whole point is that we need to get to plasmid creation, but everybody needs a little bit more background. Does anyone see how this is getting to plasmid creation, or does there need to be a little bit of an explanation about that?
Can I try to guess, or?
Yeah, go ahead.
I guess we use microscopy to detect the plasmids, and then we basically filter it out somehow. I have no idea how, but I would guess that would be the first logical step.
Almost. Well, actually, partially correct. We would use microscopy and centrifugation to isolate a special. There's a word, like important word in my opinion, called phenotype, which is like a physiological trait, like a trait that is observable or like measurable. And then you would go deeper in finding the genome for it, and we're going to attack that in later courses, of course. But it's basically from what you can see and observe and measure, you would pinpoint, and for example, having like a marker or antibody, you would find your way to the genome, like the gene of interest, and thereby gonna find the correct plasmid sequence for, or at least the plasmid backbone, and then sequence and gene and so on for compiling the whole thing. Basically, it's I still have a lot to learn, and JPL he's basically gonna teach us how to, like when we're done with the crash course, JPL is going to take over, and hopefully he's going to have like a better understanding on how to present things. And I'm also kind of trying to help him to present things by doing this as well. So we're helping each other here, and hopefully he's going to be able to tell us exactly how to look for things, how to use benchling and different tools for plasmid programming, for say, or engineering, the term is, and how to poke at different databases and so on.
Well, I have a other small question. Maybe it was already asked, so I apologize for that. But are there any interactions between plasmid and the central DNA of bacteria?
Oh yeah.
Or-
Depends on how you build it.
Okay.
Yeah.
Yeah. So, quick question for everybody who's still here. Based off of your level of understanding, do you want to learn more about how to culture and grow bacteria, because that's what you're putting, and yeast, because that's what you're putting your plasmids into? Or do you just want to go straight to the code and then you're like, I'm going to hand it off to some yeast growing guy over here?
In my situation, I would like to learn how to do the physical part myself. That would really help.
I second that.
Yeah.
Yeah.
Cool.
Personally, I'd like to have an overview on both, and yeah, mostly like having an overview would be nice to figure out where I would personally go deeper with. So yeah.
Yeah. So it sounds like the next round we should maybe focus just on micro techniques. So microbiology techniques. Sorry for the abbreviation. Micro means a lot of things to a lot of different people. Microbiology techniques and culturing techniques and some of the basics on that, because if you want to like breed some fancy tomatoes, first you need to learn how to grow a tomato, right? So we want to breed fancy bacteria. Let's learn how to grow some bacteria and yeast.
Yeah.
Well, more importantly, learn how to do it the right way. I'm a brewer and I murder my yeast at the end of every experiment. So keeping them alive would be a different story, right?
Oh, yeah, absolutely. And also making sure that they don't differentiate or kick out the genes or all the other stuff. That's a whole, that's a whole different thing right there.
I have a small question about yeast. So from what I've heard from Aniki, apparently yeast also have plasmids.
Yeah.
And small question then, can it interact with bacterial plasmids or are they different?
From what I understand, yeast is not as efficient as using with their plasmid as bacteria, because they need to, like, the mRNA need to cross the nucleic membrane, the ball at the center that holds all the DNA. It has to go through it, through the ER, endoplasmatic reticulum, and then they'll reach like the plasmids that are not as it's not as fast, basically.
But my question is, can a bacteria exchange its plasmid to a yeast, and vice versa?
Technically, horizontal gene transfer is a thing, but really, if you want to get things to work, you should be working with a backbone that is species specific.
Yeah.
Because that's how it do.
Okay, because
Otherwise things would be hot swapping all over the place.
Okay.
Yeah. Hence me presenting about the different ways of categorizing, or at least figuring out which type of cell you're dealing with, for example, and yeah.